So osmosis is the diffusion of water molecules. Okay, across a semi-permeable membrane. Okay. This might be a plasma membrane of a cell. It might be dialysis tubing. Okay. And another important component of the definition of osmosis is that there can't be a change in the pressure or volume. So if we pushed, if we had a mechanism to create more pressure on one side, it's going to if that, we're not talking about diffusion anymore here. Okay. Okay. So osmosis is the diffusion of water molecules across a selective permeable membrane without differences in pressure or volume. Okay. So we have equal volumes on either side, with equal pressures on either side of this permeable membrane. Now, this is where it gets confusing because we have been talking a lot about the concentration of solutes, but what we haven't discussed is the concentration of water molecules. Okay, so let's think about this little teeter-totter for a second. Okay. And on one side, I'm going to have my concentration of solutes. And on my other side, I'm going to have my concentration of water. If I increase my concentration of solutes, now I have to change my teeter-totter. Oops. My teeter-totter is going to be this. So I'm going to have a high concentration of solutes and a lower concentration of water. Okay. Alternatively, I could decrease my concentration of solutes, which is going to mean that my concentration of water is going to increase. Because we're talking about the movement of water, we are still going to be moving from the high concentration of water molecules to the low concentration of water molecules. Okay, the water molecules are going to move from high to low across the semi-permeable membrane. Now we might have a solute, let's call it urea. If we have a low concentration of water molecules, that means that we are going to have a high concentration of our solute. If we have a high concentration of water, that means our solute concentration is going to be low. Okay. Now, in this case, our urea cannot pass through our semi-permeable membrane. Okay. It tries to get through there and it gets boinged off. Okay. So this means, now stay with me, the net movement of water molecules is going to go from the lower concentration of the solute, which is equal to the high concentration water, to the side containing a higher concentration of the solute, which is the lower concentration of water. Okay. It can be really confusing because we're thinking about the movement from high to low, but now I'm saying it's low to high. But remember this teeter-totter analogy, and pay really, really close attention when you're reading these questions. Are you being asked about the concentration of the solute, or are you being asked about the concentration of the water? And remember that they're inversely proportional, which means that this little teeter-totter analogy. Okay. This is a potential point of confusion, so I just want to make sure that I'm spending the time here to give you guys the tools to understand that we got two things going on. We got our concentration of our solutes, and we have our concentration of our water molecules. Osmotic concentration. These terms that we're going to look at are discussing the concentration of the solutes. So we need to remember, keep that teeter-totter analogy in your mind, where we have concentration of our solutes and our concentration of our water. Okay. When we're talking about the osmotic concentration, we're talking about the concentration of all the different kinds of solutes, not just one specifically. Okay, so in your body, you're going to have potassium and sodium and glucose and amino acids and all this other stuff, right? We're talking about all of them things. Okay. In a isotonic solution, remember iso means same. And then the tonic that we're talking, which means that it gives you the clue that we're talking about osmosis, osmotic pressure, osmotic concentration. Yeah, that's how I'm gonna write that. Okay, this means that our solute concentration on the outside of the cell is going to be equal to the solute concentration on the inside of the cell. Okay, so here's our solutes. Going to be equal whether we're on the inside or outside of the cell. It's isotonic. Okay, that one's easy. So what we're going to see is our water molecules are going to have equal amounts of movement into the cell and out of the cell. So there's no net movement. So the number of water molecules on the outside of the cell is going to stay the same. The number of the water molecules on the inside is going to stay the same. In our hypertonic solution, the concentration of our solutes is greater on the outside of the cell compared to the inside of the cell, which means that our concentration of water molecules is going to be higher inside of the cell compared to outside of the cell. So now our water molecules are going to go from the concentration from high water molecule concentration to low water molecule concentration. Okay. So we see a net movement of water out of the cell. Okay. So if you drink salt water or ocean water, or a shit ton of soy sauce, you're going to get more dehydrated. Going to dehydrate your cells. In our hypotonic, the solute concentration outside of the cell is low. The solute concentration inside of the cell is high, which means that our water concentration outside of the cell is high, and our water concentration inside of the cell is low. So our net movement of water molecules is going to go from the high concentration of water molecules to the low concentration of water molecules. We're going to have the net movement. Water is going to go into the cell. I'm going to spend a little bit of time on this slide. Okay, so when our cells are in an isotonic solution, water is entering and leaving the cell at the same rate. Okay, in an isotonic solution, cells are happy. Okay. The cells are maintaining their normal shape. Everything's cool. It's all good. If we place these same cells in a condition where there's more solute concentration outside the cell. We're gonna call that a hypertonic. Okay, hyper means over. So what we're gonna do is we're gonna put our cell as the center of this, and hyper is above, right, or over. So the concentration outside the cell is higher than the concentration of solutes inside the cell. Water is going to exit the cell, and we're going to have our cell shrivel up and die. Okay, in a hypotonic, here's our cell. Hypo means below. So the concentration of solutes in a hypotonic solution is lower than the concentration of solutes in the cell. Water is going to enter the cell and cause a cell explosion. Or if you like sciencey terms, then that's cell lysis, which just means to break. Okay. That's for animal cells. If you're a plant cell, right, your isotonic solution's still gonna be happy. You place a plant cell in a hypertonic solution. We're gonna see the cell wall, the rigid cell wall is gonna stay the same, but the plasma membrane is gonna shrink and pull away from that cell wall as we see the water molecules leaving the cell. Okay. In a hypotonic, place that same plant cell in a hypotonic solution. Water is going to be entering the cell, increasing the cell volume. But again, because we have this rigid cell wall, all that extra water is going to be stored in this special organelle that I'll tell you about later when you're older. And it's going to push against the plasma membrane is going to push against that rigid cell wall and actually keep it nice and rigid. So we usually don't, so that cell wall is going to prevent explosions in our plant cells.